I want to express my sincere thanks to the organizing committee for inviting me to this wonderful forum that is addressing such a key part of what our future will be for this planet, for health, and acknowledging very gratefully the presence of Dr. Deji Tamanaga and Dean Maju Wu. Thank you for being here with us as well. It's a great honor to be here and to see so many friends from Taipei Medical University and new friends from Tohoku University. Thank you so much for this honor. I have a few disclosures for funding. My grants have come from the United Soybean Board, recent grants, United States Department of Agriculture, National Institutes of Health, from the National Institute of Aging, and the California Walnut Commission. And I just want to disclose I serve on a couple of board of directors. So we heard that I've seen estimates between 9.1 billion to 9.3 billion. But we think by 2050, our planet is going to be huge, right? We are all we know very well that our world is small, but it's huge. Knowing you all makes me feel like it's a smaller world here, and getting rid of the boundaries we have with science is one of the ways we do this. But many of the things that we need to look for, we're going to be discussing in the next 3 days. And I'm just delighted to be here and learn more about the technologies and innovation and discoveries that you all are doing to make this world prepare for 2050 and beyond. We know a key part of how we're going to sustain the world is making sure that our food systems, so using a systems approach to analyzing how food is delivered to countries that are underserved and populations in countries that are served, but have sub-pockets of people who don't get enough food that's nutritious, tasty, and of course readily available. So reducing food waste, improving farming and agriculture to protect our natural resources, and of course increasing the breadth and scale of our circular economy. You're going to hear about a lot of these topics in this forum, and I think that's going to be very important for us to think about as we look at systems approaches for the future between food and nutrition. So I have 3 sub-areas for today's talk. I'll start with what we know now, and I will do this as a storytelling from my perspective as a nutrition biologist. So my area is we look at dietary oils, how oil types in your diet will affect your health. And I'll give you an illustration. I speak my science best. I don't speak your science as well. So I used my perspective to tell this story to illustrate some of the things we're going to be seeing and talking about in the next 3 days with very many different types of approaches to our systems approach to food security for the future. Then I'll show you some of our newer findings that actually have turned to the key for how we thought of how some nutrients work. Now we're realizing just basic nutrients where we thought we knew everything we needed to know, we're learning new things about. And so I'll give you some breadth of knowledge in that area. And finally, I want to show you some ways that Ohio State is working to partner with some of the scientists here from Tohoku University, Taipei Medical University, and other universities that are attending this forum to show you what kinds of areas you'll be seeing more of in the future 3 days. So I mentioned I'm an oil chemist, an oil biologist. I love oils. I've studied them my whole career. I didn't think I'd be studying them this 30 years later, but I still am because they keep intriguing my laboratory. And we study probably the 2 simplest nutrients that are required in your diet. These are linoleic acid and alpha-linolenic acid. So you know we need vitamins, vitamin A, vitamin C, vitamin D. We have RDA set for those in the United States. We also need these 2 nutrients to the same extent that we have requirements for vitamins, but unfortunately there has not been enough research on these 2 nutrients to set up RDAs. So what we are learning is how we're going to set these RDAs up so that we not only prevent deficiency, but maybe prevent some of the chronic diseases that we think are also associated with shortages of these 2 nutrients. Now, why am I talking about 2 fatty acids? As I mentioned, they're very simple molecules. You don't need a lot of chemistry to see this molecule is an 18-carbon molecule. It has 2 double bonds, so 2 desaturated tetracarbons, and 1 carboxylic acid. That's really all fatty acids are. These two, this is an omega-6 fatty acid and this is an omega-3 fatty acids, again, are the 2 only required fatty acids in the adult diet that we think. They are also polyunsaturated, so there's 3 double bonds in this omega-3 and 2 in the omega-6. And we know we have requirements, but we, again, in the United States don't have an RDA yet, so we have what we call adequate intakes, between 11 grams to 17 grams for linoleic acid, and alpha-linolenic acid between 1.1 to 1.6 grams, so about 1/10 lower. And I'm pointing out these amounts here because I'll show you some fortification studies where we use a modest amount of fortifying oils in the diet of people with diabetes or sarcopenia, or we have a new study that I'll show you about people with non-alcoholic fatty liver disease where we use modest amounts to boost up the levels of linoleic acid to show how that can change how they respond to their metabolic diseases. And it's quite profound and like a lot of things in science, was very unexpected when we started looking into this more. This one last structure thing that I want to point out to you is this is a phospholipid. There's a phospholipid head group, a side group, and then the 2 fatty acids. And the reason I'm showing you that is many people think of oils and fats as being calories, and they are. There's lots of energy in these carbons, right? You break 2 carbons off at a time of fatty acid through beta oxidation, you produce a lot of ATP in the body. But in polyunsaturated fatty acids, they really prefer to go into phospholipids, not triglycerides. So triglycerides are that glycerol form of fatty acids that serve for energy storage. Phospholipids are what line all of your membranes of your cells and all of the organelles within your cells. And what my lab looks at is phospholipids in the mitochondria to see how these particular fatty acids can fortify the mitochondria to work better for diseases like sarcopenia, type 2 diabetes, et cetera. So we have looked in my lab for most of my career at chronic conditions that we used to think were only in our older population. And probably like the United States, I think Japan and Taiwan have the same problem where we're seeing a graying of our population. People aren't having as many children, and so the average age is creeping up. And these chronic conditions are also increasing to the point where I believe they're an epidemic on our medical care system and also, of course, for patients' quality of life and lifespan. So I mentioned we're going to talk about what we already know about fatty acids. And this was published in 1957 in the American Heart Association journal Circulation. The American Heart Association published this position paper reminding physicians and scientists that vegetable oils that decrease cholesterol is thought largely due to a component known as linoleic acid. So for 70 years, we have thought, and we continue to see evidence for, I'm not touching anything, I promise. We have seen evidence for a protective role of this fatty acid in heart disease. So how many of you, just raising your hand because participation helps keep you awake, how many of you know that linoleic acid is protective for heart disease? So I don't see a lot of hands, and that's okay. It's the same thing in the United States. People don't know this, and it still continues to be shown in epidemiological studies and animal studies and randomized controlled trials that all of the risk factors for heart disease, including the event of having a heart attack and including deaths from heart attacks, are lower in people with higher intake and blood levels of linoleic acid. But we're not very good at getting that out in the United States either, so don't worry. So this was 70 years old, and I'm going to show you even a recent paper that still shows the same thing from actually the same journal, but doesn't seem to be making an impact yet in recommendations in the United States. A while ago, so this is now about 14 years ago, so this is not I should have said new and almost new studies. About 14 years ago, we were working with a dietary supplement that's sold in the United States, and it might be sold in Japan and Taiwan, I'm not sure, that is composed of a fatty acid called conjugated linoleic acid, or CLA. And CLA is also marketed under the brand Tonalin as a weight loss supplement. I don't know about you all knowing this, but in the US, weight loss supplements rarely actually have any evidence to show that they cause weight loss. More of it is people think they lose weight and they might lose weight. So we wanted to test whether this dietary supplement could actually help people with type 2 diabetes who were obese lose body fatness. And so we were testing this in what's called a randomized double-blind crossover study. So we were comparing the CLA to safflower oil. Safflower oil is a common vegetable oil in the United States, very rich in linoleic acid, traditionally. And so we had a 36-week intervention here, 2 16-week diet periods, and we were working with women with type 2 diabetes. I think the connection is maybe loose or something, but we were recruiting women with type 2 diabetes who were obese. And so we recruited 55 postmenopausal women. Average age was 58, BMI was over 35, so everybody actually was at least overweight, if not obese. And many of the women had had diabetes for at least 10 years, many more than that, with a variety of oral medications to control their type 2 diabetes. So a variety of oral medications, but what we were doing in this study is supplementing them with 8 grams of CLA, which is marketed and was marketed then even as Tonalin, and then comparing 8 grams of CLA to 8 grams of linoleic acid-rich safflower oil. 8 grams is less than a serving, at least in the United States, and so it's to give you a sense of how much. This was less than a teaspoon and a half if you use teaspoons, or of course 8 grams, serving. So again, this was 16 weeks diet period, and then the women were taken off of whatever supplement they were on, either the Tonalin or the linoleic acid, safflower oil, and then moved to the other for the second diet period after a 4-week washout. And what I'm showing you here was published in 2011, so it's an older study, but it is illustrating some new findings. This particular graph is not new. It was showing that the linoleic acid-rich safflower oil increased HDL, which had been shown, and again, that would be associated with that linoleic acid protection against heart disease risk by raising the good HDL, the good cholesterol lipoprotein fraction. And the CLA actually had no significant effect. It didn't significantly lower it in this clinical trial on average between week 0 and 16. The asterisk here means the difference, the delta between week 0 and 16 in the women was significantly different. And then this p-value means that the 2 treatment groups were having a significantly different effect. Okay, so the effect was different when women were on linoleic acid or CLA. The next several slides have the same kind of annotation. So when we looked at glucose, we hypothesized, the main hypothesis in the study was CLA would reduce body fat and help lower glycemia, maybe to help women not have to take as many oral medications in the long run. We didn't know. But that's not what we found. You can see CLA had no effect on either fasting glucose or HbA1c, which is the long-term measure of glycemic control. And we also, as I mentioned, we looked at 2 different markers of glycemic control at week 0 and 16. And again, significant reduction by safflower oil, which was not meant to be having any effects in this study. So if you're a scientist, which I think all of us are from some area of science, you know most of your experiments don't go the way you think they're going to go, but usually they don't go the opposite of what you think they're going to go. But in this case, they did. So that was a whole nother learning lesson for me. And so you can see safflower had a significant effect on lowering glycemic measurements by fasting glucose or HbA1c, as shown by the asterisk, and then the p-values were significantly different effects in each group for each of those measurements. We also measured body composition at the beginning and ending of each diet period in this crossover study, so week 0 and 16. You don't think people are going to show in 16 weeks, which is about 4 months, huge effects on body weight. And in fact, though, we did see an effect on body weight by the CLA, which was kind of supporting the idea that they are weight loss, but there's other issues with that. But I'm going to show you the body composition because we thought this would be more sensitive to findings in that short period of time, being 4 months. And unexpectedly, what we found was safflower oil reduced trunk adipose mass measured by DEXA by about 3.5 pounds, or you can see to about 1.5 kilograms. And then CLA had no effect on trunk fat. We had hoped CLA would, because again, that's the dietary supplement promoted for weight loss supplement, had no effect. Whereas the safflower, which was supposed to be a neutral oil in this study, had a significant lowering effect of trunk fat. So again, we were kind of scratching our heads on why this could be and how this happened, and are we the only study or only lab that showed this? Turns out we're not, but this was definitive at that point for our study. We also looked at a hormone called adiponectin. So adiponectin is a cytokine that's produced from adipose, so adipo and nectin. It's a cytokine that's actually a beneficial cytokine. So we were thinking that the CLA would increase adiponectin, and it didn't in this study. And then again, unexpectedly, the safflower oil rich in linoleic acid increased adiponectin in these postmenopausal women. The increase was significant here. You can see the p-value was not significant between the group effects. So there was not a significance between CLA and linoleic acid here. So adiponectin, as I mentioned, is a cytokine mostly produced from adipose. Since the discovery of adiponectin, people have found it can be secreted from fatty liver, muscle that has some fat in it, and other organs. So it's produced from tissues that have fat storage. And it's still associated, if you have higher levels, it's associated with better insulin sensitivity. So it's secreted mostly from adipose, that's the primary source in the blood. And the protein causes lower glucose output from the liver, lower fat accumulation, lower inflammation, can increase energy expenditure in muscle, and can reduce, again, inflammation associated with heart muscle. And so adiponectin is thought to be a protective cytokine. So we were intrigued by this finding. That had not been shown prior to our study. And we were intrigued because adiponectin is a PPAR gamma responsive gene. So PPAR stands for peroxisome proliferator activated receptor, which is a mouthful, but it's a nuclear hormone receptor. To give you a sense of what PPAR is related to, estrogen receptors are also nuclear hormone receptors. So they work very similarly. Estrogen receptor binds to estrogen, or estradiol. PPARs bind to fatty acids and fatty acid metabolites. It is also associated, PPAR gamma activity, with insulin sensitivity. So there was a whole group of drugs that were targeted and developed and used for many years called thiazolidine diones. And you had them here because actually one of the companies that was a big producer was from Japan. They were very effective at insulin sensitizing people with type 2 diabetes. They had such a potent effect, though, that they also caused side effects, so they were removed from the market because they're slow to metabolize, the drugs are. Fatty acids, though, are short-living compounds that bind to PPAR gamma. And one of the ways that PPAR gamma may be insulin sensitizing is by increasing adiponectin gene expression, which would then result in increased adiponectin protein translation, of course, and then secretion. When adiponectin is secreted from adipose as a protein, it goes to target tissues and helps those tissues respond to glucose better to metabolize glucose and fats for energy. So that's how it works to sensitize energy, sensitize the body to insulin. So this is important because of the effects we saw on linoleic acid, showing that it reduced glycemia, which is what you expect with insulin sensitizing, and it increases insulin sensitizing hormone. So we thought this could be a mechanism that we should pursue. And that's a review of adiponectin. I'll jump through this one. So we designed a smaller study to try to prove of principle to see if we could show another, because that was the first study to show adiponectin increased in people after linoleic acid supplementation. We wanted to show it again in a less sick group, meaning less sick. They weren't women with type 2 diabetes. They did have metabolic syndrome, though, so they were starting to show signs that they could be developing type 2 diabetes. So these women, we recruited 15 women. I could not get this grant funded to save my life. It took me too many trials before I gave up and decided I would put together a creative budget. If you're an administrator, don't listen to that. But creative budget, meaning I was an endowed professor, so I had a little bit of funding to do things like this. And we developed a single arm trial, so there's not a comparative arm, There can't be blindness then, right? You do know what the women are being treated with because there's only one treatment group. And we had a very small cohort, just 15 women. All of them were overweight, but they weren't quite as obese as the women in the women's diabetes study that we had. We designed the study fairly similarly in that the timeline was 16 weeks for the diet period. Every 4 weeks, we met with the women, replenished their oil supplements, took blood glucose measurements, as well as measured body weight. We did not have enough funding to do DEXAs in this study. And we published a study, and we did find that after 4 weeks, adiponectin levels increased in the blood significantly, and they were maintained throughout the 16 weeks, which was interesting. We hadn't done a time course. We also did find in this study that adiponectin at the high molecular weight form, which is the bioactive form, was increased. So we thought this could, again, be explaining some of the glycemic improvements that we saw in the women's diabetes study, but we haven't made that direct cause-effect relationship yet, so these are correlations, not causation yet. Oops. What we also were able to do in that study is do some targeted lipidomics, and you're going to have some talks on targeted lipidomics in this forum. So I wanted to show these data because from a very simple supplementation with an oil in a pill form, then that's linoleic acid right up there. This is a network analysis showing all the metabolites that are increased, which is in red, and the metabolites that are decreased, which is in blue. This is the omega-6 pathway between linoleic acid and arachidonic acid. And then I showed the omega-3 pathway because some of those metabolites also increased from the linoleic acid supplementation, which isn't what you hear about in media in the United States. There's a lot of people who say omega-6 fatty acids are bad because they decrease these guys. And it's not what we find ever in animal studies or human studies, but there's a lot of myths that get perpetuated, at least on media in the United States, and I'm guessing here as well. So we do see that there are increases in some of these omega-3 metabolites. Importantly, we also do not see, after 8 grams of supplement of safflower oil with linoleic acid, that there's any increase in arachidonic acid. That, again, has been shown for 30-plus years, because I showed that in my PhD dissertation, and I still passed my doctorate defense, by the way. Students, if you're in this room, if you find negative data or even opposing data, you will still pass, because that's important, right? So we've showed that in a mouse model for skin cancer, and I've continued to find that in all of our studies. This was a collaboration with John Newman out at USDA Center in Davis, California. Excellent lipidomics expert in the oxylipin field, if you're interested. And so he was able to help us with not only the chemical analysis, but the network analysis, so we could really understand what's changing that could be important. Now, what I want to tell you is several of these oxylipin metabolites are extremely high affinity ligands for a receptor, PPAR gamma. So now we're making a closer link between what linoleic acid did with glycemia, PPAR gamma activity, adiponectin production, and possibly insulin sensitization. Again, all of that is associative because it's really hard to get a causal effect in a clinical trial, but we're creeping closer. When we looked back in the literature to write up these papers with this very unexpected finding, I kind of kicked myself because I was not aware that there had been several studies conducted in Sweden at this point. This was Bengt Vesby's group. He's now since retired, but he led up a group in Sweden where they looked at linoleic acid and its importance in insulin sensitivity. Again, different than heart disease protection, right? Insulin sensitivity is associated with diabetes. And he had found that when he measured insulin sensitivity with a very sensitive method called the hyperinsulinemic euglycemic clamp, in older men who were not diabetic but insulin resistant, we all get more insulin resistant, unfortunately, as we get older, that serum linoleic acid is positively associated with better insulin sensitivity, so better insulin effects when they had higher linoleic acid. What brings up your linoleic acid? Only diet, because you don't make it in your body, right? It's an essential nutrient. So when people show biomarkers of linoleic acid, they're implying that it's from the diet because you only get it from the diet. But he didn't look at diet in this study. And then a study did do a supplementation study and looked at improvement of insulin sensitivity in people with or without diabetes. So this was a paper done by Kevin Frain's group and published in 2002 showing that when, in a crossover study, when people with insulin resistance or type 2 diabetes were given saturated fat food products for 5 weeks only, it shows a significant reduction of insulin sensitivity versus when they were given a linoleic acid rich group of foods for 5 weeks. At the end of it, the hyperinsulinemic euglycemic clamp showed higher levels of insulin sensitivity after the linoleic acid fat-fed group. So again, showing that there could be a true cause effect between linoleic acid and insulin sensitivity and what we found in the women with type 2 diabetes. There's now been many other studies. This was one of the more recent ones published in Nutrients showing that linoleic acid, which is up here in the top left corner, was associated with reduced risk for type 2 diabetes and was associated with better glucose. So low percentage of vegetable fat, being linoleic acid in this graph's case, was associated with, so lower linoleic acid in quintiles here, associated with worse odds ratio for type 2 diabetes and for worse glucose control. Alpha-linolenic acid started to show similar things at the higher quintiles of alpha-linolenic acid. No associations with the two long-chain omega-3s. In fact, DHA, and this is not the first time, but higher levels of this particular long-chain omega-3 were associated with worsened risk for type 2 diabetes and glucose metabolism. So just note that it's not as simple as we thought, right? What we think we know is sometimes not what actually is true in science. And I mentioned that the other thing my lab does is we look at then how fatty acids get into phospholipids and change how that cell responds to the environment around it. And one of the main ways we do this is we look at mitochondria, which are rich in membranes, right? There's an outer mitochondrial membrane, there's an inner mitochondrial membrane that expands to form bigger cristae when the mitochondria need to have more energy production for beta oxidation. Your cristae get more proliferative in the mitochondria. And it turns out that the inner mitochondrial membrane the inner mitochondrial membrane prefers linoleic acid as its fatty acid component. It's the only fatty acid it prefers. So other fatty acids will get into this inner mitochondrial membrane phospholipid called cardiolipin. But if that's true, then the mitochondria don't work as well to make ATP. So this is a molecule called tetralinoleal cardiolipin. And it's a phospholipid only found in the inner mitochondrial membrane. You don't see that phospholipid in other organelles. So it's very specific to forming cristae and helping the cristae support the electron transport chain. So, and this is just showing you again, depicting that the cardiolipin is in this inner mitochondrial membrane that form these nice cristae to support more surface area, to support more electron transport chain machinery, to make ATP. One of my collaborators in this group, another lipidomics expert who's in Colorado, has written many articles on cardiolipin, and she has taught my lab a lot about it. We collaborate pretty closely on this work because I'm not an energetics biologist. And what she has said is what seems to be true about cardiolipin is it forms a three-dimensional structure that's optimal to pull the electron transport chain complexes together. So there's 5 complexes, five main complexes, right? 1, 2, 3, 4, and 5. And those have to be pulled together to allow for electrons to be passed from one complex to the next to maintain this hydrogen gradient, which allows for then the subsequent production of ATP through complex 5. And if you have leakage in this, you have inefficient ATP synthesis, which causes all sorts of other problems in the cell. That happens in cells when cells undergo cell death. There's a lot of leakage of electrons. The electron-proton gradient can't be maintained, and mitochondria start to break down. That's a whole new process that we've learned about called mitophagy. So we think linoleic acid is important for maintaining healthy inner mitochondrial membrane for energy production. And that would be important in things like managing type 2 diabetes and energetics for obesity and sarcopenia and fatty liver disease. And that's kind of where my lab started to move toward in the last 10 years. So 2 people in my lab, Dr. Cole, who's currently still my lab director, and then a prior PhD student named Austin Angelotti, worked on a very short-term study. This was another study that was creatively funded with some funding that I was able to get internally at the university. And we did a 2-week intervention with either a linoleic acid-rich cookie or an oleic acid-rich cookie. And both oils were from safflower oil, so we now have high oleic acid vegetable oils in the United States, and I think those are common in other countries too. They're more shelf-stable, more stable in a fryer. They're better for diesel engines too, for tractors and things like that. I mean, they're important. So these oleic acid-rich oils are now in the edible form in our markets, but there's still the old-fashioned form of safflower with linoleic acid-rich. So you can see the composition. The linoleic acid-rich oil had about 9 grams of linoleic acid, and the oleic acid-rich just had the same amount, 9 grams, but of oleic acid instead. And we recruited healthy participants in this study to consume 1 cookie a day. Many of them wanted to consume more, but we didn't want them to consume more than 1 serving of oil a day that was fortifying their bodies and their diets with linoleic acid or oleic acid. I'm going to skip through this, but this was a healthy population to see, and there were 42 people in either diet treatment arm. Little over half were females. You can see ages were younger than most of our cohorts. We did recruiting a lot around campus for people pursuing their degrees or their graduate degrees. And what we found is this graph on the left is showing you linoleic acid levels and oleic acid levels of either women on healthy participants on the linoleic acid-rich cookie, which shows you here there was about a 2.5% increase, and that's unit percent increase in the blood, and percentage is how you measure these fatty acids, so it's a unit, or a decrease of oleic acid in the people fortified with linoleic acid cookie. The oleic acid-rich cookie had no effect on either of these fatty acids in the blood. But when we looked at tetralinoleal cardiolipin, so this is that linoleic acid-rich cardiolipin that's important in optimal mitochondria function, we found that the linoleic acid cookie, just with 9 grams a day, was able to significantly increase cardiolipin. And this was in PBMCs of healthy adults. Why is this important? Because it's never been shown that we know of still in published literature that you can fortify a diet with 1 serving of a certain oil and increase mitochondria cardiolipin, tetralinoleal cardiolipin, this cardiolipin that's so important for health. So we're now trying to repeat this study where we can then measure in those PBMCs mitochondrial function using oxygen consumption measurements, and those are studies that are currently ongoing. So the linoleic acid-rich cardiolipin increased in the linoleic acid oil cookie-fed people, and some of the linoleic acid-poor cardiolipins were reduced, which is kind of what you would want to see. So we are working on this kind of lipokinetic action of linoleic acid in 2 ways. We're looking at linoleic acid-rich oils increasing the electron transport chain to improve glycemia, and we're still pursuing how linoleic acid is a precursor for these oxylipins to perhaps stimulate PPAR gamma to also improve glycemia, and those studies are ongoing. One of the studies I wanted to show you today was a new study where we're looking, and we just wrapped up the final participants visit in December, which seems like yesterday, but that's a long time ago now. But anyway, and we're finally getting our data analyzed. We're analyzing our data. One of the things we wanted to do in this study was, again, look at linoleic acid fortification of the diet to see if people with non-alcoholic fatty liver disease could show improvements in their liver lipid levels, which is a problem in many people with central obesity and metabolic syndrome. And so again, the way we measure the metabolic syndrome is by measuring these proponents of metabolic syndrome: central obesity, dyslipidemia, insulin resistance, hypertension. And I'll show you some of these data. to understand what non-alcoholic fatty liver disease is, which, by the way, now the AASLD, which is the American Association of the Study of Liver Diseases, they have now decided that most non-alcoholic fatty liver disease is actually metabolic-associated steatosis of liver diseases. So now they've renamed it MASLD, just to confuse us more. But it is more appropriate. It is more closely linked with what people with non-alcoholic fatty liver disease really have, which is the manifestation of metabolic syndrome in the liver. Unfortunately, between 20 and 30% in the US have NAFLD or MASLD. Because of our obesity rates being so high, this is really becoming a commonly diagnosed disease. We don't know how many because it does take quite sophisticated measurements to actually really assess and determine if someone has it. Children with obesity are also now developing NAFLD or MASLD, which is hugely problematic because once you start messing with the liver, that's the central place that really is what's really directing a lot of metabolism in your whole body, right? So we're very concerned about this in the United States. So in this study, and this was the study that was funded by the United Soybean Board, we fortified people's diets with either soybean oil or control oil for 16 weeks. We used the same duration as the women's diabetes study again so that we could do some comparisons, and we know that's long enough to see some of the changes in body composition. Again, all of these people were people with non-alcoholic fatty liver disease, so they needed to have a diagnosis from a hepatologist in our internal medicine group at Ohio State University. We measured liver fat with magnetic resonance imaging and body composition with DEXA. We have dietary assessment measurements throughout the study, but we haven't finished quality controlling or QCing all the data and analyzing it, so I can't show that to you today. But we have fasting blood levels of some endpoints that I'll show you today. And we also looked at changes of medications. So you always think you have the whole study figured out ahead of time. How many people in here are researchers? Is everybody here a researcher? So you know you've done the experiment before, you think, okay, no problems, we've done it now, we've worked out all the kinks in the last study. But we forgot that in the United States, that's just a MRI image. A lot of people are going on these GLP-1 agonists. I don't know if that's happening in Japan or Taiwan. Is that happening? For diabetes control was what they were developed for, but a lot of people in the US are going on it for weight loss. So in our cohort of 60-something people, 78 people actually, we had 16 people on GLP-1 agonists. That's a lot of people. Yes, they're all people with non-alcoholic fatty liver disease, so they're all overweight or obese. Not all, but most were overweight or obese. But still, we didn't anticipate that, and we didn't soon enough predict it to know we should get rid of that or put that in our exclusion criteria. So this was a weight-neutral study where we wanted to fortify diets with soybean oil or control oil and see if the soybean oil had an effect on fatty liver, right? We were hoping they wouldn't gain or lose weight because that will affect their fat in their liver. And so this kind of is a confounder that we're now going to have to include in our data analysis, which unfortunately changes our power, but we'll have to handle it the way we have to handle it. So we had 28 men, 42 women, and 4 people who identified as neither man or woman as sex. BMI was average of 36, but we did have few people under 30 even for BMI. Fasting glucose was within normal range, so these people were mostly not with type 2 diabetes, although we did include a few people with diabetes if they were well managed, showing us their glycemic HbA1c levels, HDL, CRP, MRI of fat percent. This is showing you all of them had fatty liver. This would be about 5% or less if you don't have non-alcoholic fatty liver disease. So they all had fatty liver, as was a qualification to enter the study. So here's what I'm showing you about the GLP-1 agonists and what they did. We were supposed to see no weight changes, or if people were going to undergo weight loss, we counseled them. We worked with them, by the way, every 2 weeks with a registered dietitian to help them incorporate the foods to hopefully keep them weight neutral. But you can see some people lost within 16 weeks. One person lost, 15 kilograms of body weight. I mean, these people might have been large to start with, but that's still a pretty significant weight loss for being weight neutral. So we had a lot of people lose weight. We had a few people gain weight. There was no group difference between the weight changes. We also divided each group by sex, and you can see there's basically no difference between men and women for weight change. Just quite variable, and again, not what we were hoping for, but that's the way it goes. This was our primary finding, change of fat fraction of the liver measured by magnetic resonance imaging. And what we found was the treatment groups were significantly different in how the people responded. So the people in group B are on the soybean oil, which is rich in linoleic acid, and we saw a significant difference between how soybean affected fat versus the control oil, which was palm oil in this case. This is important because this was our primary hypothesis. We did not see a significant reduction of fat within the treatment group, though, and I want you to note that. We have not adjusted the data for GLP-1 use in either of the groups yet, and so that's one of the things we're going to be doing in future data analysis that might explain some of the variability within this graph. But that's where we are at the point of today. We haven't published any of this yet, so this is brand new. We looked at plasma levels of linoleic acid to see if people were complying to eating the linoleic acid fortified foods. And you can see most people increased their linoleic acid levels. Some people did not. I don't know if they were not adherent. We also have logs, but people aren't as good about keeping logs. But we are still going to go back and see if we can identify if these people were absolutely not compliers. But they'd come to our study visits and tell us they were complying. Otherwise, they had to be stopped from the study. So, people don't always remember or something. And then alpha-linolenic acid, which is also another fatty acid in soybean oil, and remember that's an essential fatty acid that you need in your diet, did increase in the soybean group, and it decreased in the control group, the palm oil group. You may wonder why we chose palm oil. Does anybody wonder why we did? It's actually very representative of the PUFA, MUFA, saturated fat distribution in the typical American diet. So we chose it to not change fatty acids, although you can see it did reduce alpha-linolenic acid here, but we wanted it not to have an effect. You always want your control group to be as neutral as possible. That didn't work for me in the women's diabetes study, just remember that, but we try for that. And so that's why we chose the palm oil as our control oil, and it did have an effect on alpha-linolenic acid levels. We saw also that arachidonic acid, once again, did not increase in people fortified with linoleic acid. That's the common myth that linoleic acid, the seed oils, vegetable oils, whatever you want to call them, there's this whole new thing about seed oils being bad for you now in the United States. I don't know where it's coming from, but it's because they think it increases arachidonic acid, which increases some pro-inflammatory cytokines. But we never see arachidonic acid levels budge. We didn't see it significantly change in this linoleic acid fortified group either. It did show looks like a decrease, but it wasn't significant. And we never see a decrease in this long-chain omega-3 fatty acid, EPA. So again, another myth that if you fortify with vegetable oils, you're going to harm omega-3 levels. It doesn't happen in people. It doesn't happen in tissues of animals either. So I don't know what to say, except it just keeps popping up in the media. We looked at many markers of inflammation, and this is LBP, which is a receptor for endotoxin, which could be a measure of leaky gut. We didn't see any effects of either group, treatment groups on LBP or oxidized LDL either. So at this point, from the WELL study, which is the weight and energy for lipids in the liver, we are finding that there could be some impact of soybean oil or linoleic acid-rich oils for reducing fat in the liver of patients with non-alcoholic fatty liver disease. We don't see an increase in arachidonic acid, but we did see changes in LA and ALA. And some of these markers that we would think are deleterious for people with non-alcoholic fatty liver disease didn't change. There may also be sex-specific effects of soybean oil. I didn't go into too much of that in the previous slides, but we're going to continue to investigate that more. And then finally, of course, unexpectedly, the GLP-1 agonist drug use that is happening and being prescribed in the United States for weight loss and diabetes management did have a confounding effect that we didn't anticipate when we set up the study. And that's the way research goes. So finally, my last third of my talk, which is not a third, it's shorter. So hang in there. Am I over time? Okay. So extending nutrition research to food science for the future. I am delighted that I got to be, for just currently, the department chair of the food science department at Ohio State University, which really has not historically had a lot of nutrition in it. Nutrition moved out of that about 40 years ago and moved to another college at our university, unfortunately for nutrition, because it's not doing so well over there. But food science is starting to integrate nutrition back because of what we heard this morning from our president and dean, that interdisciplinary food and nutrition is needed for addressing the problems that we could be facing in the next 30 years and 100 years and hundreds of years for our population growth. And so some of the things that we're doing in nutrition, again, I wanted to use my research as just one way to illustrate how things are changing. We found in my women's diabetes study, and other people had shown this too, that linoleic acid increases in the blood were associated with reduced risk for diabetes and better insulin sensitivity. I showed you some of those data from some other labs as well. This was a very large meta-analysis done by a very large group of epidemiologists from all over the world, where they combined studies from, how many studies? Was it 30 studies? 20 prospective studies with almost 40,000 adults from all over the world. And why is it all over the world? Why is that important? Not only does it show global collaboration, which is what's needed in a food systems approach, but it's linoleic acid sources from all sorts of different types of oils. In the United States, our primary sources are soybean oil, canola, safflower, sunflower. In desert countries, safflower is one of the main sources of polyunsaturated fat oils. In other countries, it could be other types of oils. So importantly, wherever the linoleic acid came from, higher quintiles of linoleic acid were associated with a significant reduction of relative risk for type 2 diabetes. This is 0.57 relative risk compared to the lowest quintile of linoleic acid in the blood. That is huge. That is huge, right? So higher levels in the blood, lower risk for type 2 diabetes. So far, that's not being picked up on as a dietary recommendation by the American Diabetes Association. I don't know if it's being picked up in other countries yet. And because of so many people in this cohort, they were able to adjust for a number of different things, but including age, BMI, sex, race, omega-3 polyunsaturated fat levels in the blood, aspirin use, and then this polymorphism called fatty acid desaturase polymorphism. And what you can also see here is arachidonic acid levels, which some people say promote inflammation because of the eicosanoids, actually had no association with type 2 diabetes risk at all. So again, arachidonic acid isn't quite the bad boy that, or the villain that people want to make it. So I study food as medicine, and I want to show you a paper that came out in Circulation. So in the beginning of my talk, I showed you a paper from 1952, was it? I can't remember now. 1950s, that was before I was born. I just know that much. Now this is a newer paper, 2017, from Circulation, same American Heart Association journal, from the Presidential Advisory Board. And this is in the abstract, but then it's shown in the data that are in this paper, and it's a really fun paper to read if you're interested. And I'm not an epidemiologist, but I can understand it, so that says something. Polyunsaturated vegetable oil reduced cardiovascular disease by about 30%, similar to the reduction achieved by statin treatment. So statins are the #1 heart disease drug prescribed in the United States. #1. I don't know what it is in Japan. Is it pretty high? Maybe not as high. I don't think dyslipidemia is as prevalent, but I know it's not trivial here either. So we're talking about an oil that you can buy in the grocery store that doesn't even have to be in a pill, that can have the same level of reducing risk for heart disease as statin, which is the #1 drug that many doctors think is the miracle drug, right? How many people know that this could happen, and that increasing vegetable oil just maybe by a serving or so a day could have an effect on lowering risk for heart disease and heart attacks and heart deaths, which were also shown in the studies that were cited in this paper? I don't know. A lot of work to do. So, but foods have medicine properties, and we need to remember that, even common foods like vegetable oils. This is just showing you from the Lancet diabetes paper that I showed you with a reduction of diabetes risk with linoleic acid. This is showing you the curve, and this is showing you in our study what we found with 10 grams of oil a day in our papers, how much that could change plasma levels. So you can see how that could have an effect in the curve that is shown in this paper, just to kind of give you a possible translation back. Okay. So just food for thought, how many adults are being prescribed linoleic acid-rich vegetable oils or seeing a dietitian? I think in Taiwan, I learned last year, Taipei Medical University, and in a conference I was attending that I was just lucky enough to be attending, dietitians are more used in medical care than they are in the United States. We have a long ways to go for that in the United States, and I'm not sure, I hope in Japan dietitians are used, because people need help with their diets to get some simple incorporations of foods that could be very valuable as medicine. So why are we interested in vegetable oils? Well, one is we think if we could add just a little bit more to some people's diets who might need it, they could have benefits for diabetes, heart disease risk reduction, sarcopenia, which is an ongoing study we're doing right now, and maybe even people with fatty liver disease. But the other reason is we know oils in the market and in foods are changing. And this is in the United States. I don't know if we have any people in here who study oils. And I'd like to know if it's happening in Japan or Taiwan. But many of our vegetable oils are becoming high oleic acid sources. And I don't know if that's happening even in Europe. I think it is. And I mentioned this before, but the reason is they're more shelf stable because they only have one double bond versus two. They're more shelf stable. They're more stable in a fryer, so they don't oxidize as quickly, so people don't have to change their fryer oil as frequently. They're more stable for diesel engines, so they don't get sticky when you use vegetable oils in some diesel engines that are currently being used in the United States. So they're good. High oleic acid oils are good. The other thing is they form less trans fats, which is that hydrogenation process that forms these trans fats that are not good for heart health and probably not other diseases associated with chronic disease risk. And so high oleic acids have a place in our edible oil supply, but what's happening, and I was with a group who did some mathematical modeling, what's happening is if those become more than 40% of our oil supply in the United States, we're going to see inadequate intakes of linoleic acid to the point that we're almost going to see deficiencies. So this happens a lot in food supplies for people, but we see a lot of pendulum swinging where one thing gets solved and another problem pops up, right? We call that sometimes, I don't know what we call it, I lost my train of thought there, but we don't want that to happen. So we need to be cognizant of the whole system of oils as part of our healthy diets. Then there was also a study that was just published this year in the American Journal of Clinical Nutrition, another mathematical modeling study showing that with climate change and the warming of our globe, so on the x-axis down here is higher levels of mean high temperatures in different latitudes, and this was done in North America between Canada and the United States. Linoleic acid levels are going to go down. Alpha-linolenic acids are going to go down in those oils. So now we're looking at other ways that people may not be meeting their essential fatty acid intake. By the way, oleic acid levels go up in these oils when we see higher mean temperatures. And I'm not picking on oleic acid. That's a monounsaturated fatty acid. It's not essential. People in the United States get about 45% of their fat calories from oleic acid, so we're not needing a lot more of it. We don't need any more of it. But it's just not helpful for what we think linoleic acid is important for. What about food safety, security, and authenticity? These are things that you're going to hear about in the next 3 days, and I want to kind of shift gears on this because we have a couple of people, Dr. Ahmed Youssef at The Ohio State University, who's developed some techniques to identify antimicrobial peptides produced from Brevibacillus species of bacteria, and he's done a lot of work in this area, and we're going to hear from Dr. Ryuto Toby. Is he somewhere here, hopefully? who's going to be talking about his work on antimicrobial peptides against pathogenic bacteria. This use of basically battling microbes against microbes is really going to, I think, take off as we understand more about the microbiota and how that's important in helping microbiome populations work together to maintain health and to keep our foods safe as well, and packaging. We have a researcher in my department whose name is Dr. Luis Rodriguez-Sayona, who uses handheld FT-NIR spectroscopy for measuring authenticity of foods as well as components of foods that could be altered by either processing, growth, or sometimes, unfortunately, intentionally. There was a big olive oil scam, I guess, where some olive oils weren't full of the true olive oil components that they were supposed to be. And Dr. Rodriguez-Sayona was one of the people who was involved in some of those studies. Using these handheld spectroscopic techniques can allow for real-time on-site data collection where data can be collected and integrated into a systems approach as we continue to talk about the need for food identification as well as food nutritional aspects and taste for health. Harnessing the power of microorganisms for cardiometabolic diseases is the theme of the next few sessions for today, for the rest of the day, I think, and some of the sessions coming up tomorrow and Saturday. And so you'll hear about some sessions looking at microbiota populations for gut health. I know we have a few speakers talking about secretory metabolomics from microorganisms. So lipidome, I know we have one speaker on the lipidome, as well as microbial genesis of bioactives that can be useful for food ingredient making and even making packaging, which I think we'll hear about at this meeting. We have someone in my department using spent coffee grounds to do this kind of packaging development, which I think is exciting. Just one feature is Dr. Nobuyuki Okahashi is going to be showing you some work where he uses tracer stable isotope measurements to understand metabolic flux. And he's using this in identifying metabolomic and lipidomics of microbiome secreted products. And I think that's going to be exciting, but we have several other studies that are being presented by many of you. So I'm excited to see this work. And to infinity and beyond. Finally, this is my final piece, and I'll end within the next few minutes. That's a quote from a very famous guy called Buzz Lightyear, Toy Story. We were fortunate enough to host a series of companies at my department this past summer who were using our pilot plant, our food production pilot plant, to test different technologies that could be used in long-term space living. Why is that important for us on Earth? It's pretty obvious to all of us. Some of those techniques are going to be used, and some of them are going to be discussed in this forum. Things like what I would call tricking photosynthesis to help release oxygen in a way that's not as dependent on photosynthetic light waves as needed for producing oxygen. We're going to hear some of that today, and there was a company visiting our department doing that same kind of work. And so NASA came to our department along with funding from the Methuselah Foundation that went to these 4 companies, and these are 4 private companies that we had nothing to do with choosing. They were chosen by the NASA judges, sent to our labs for the summer, and then we had 4 what we called Simunauts, which are food science students who worked with these companies so that companies could test how doable their technologies were for people who are not trained in the food science field to make sure that the foods could be produced with safety, taste good, and of course, nutrition. The staff of, and I should have had a photo of the staff from the pilot plant. Our staff are Matt Papik, Stephen Simmons, and Matt Crucial. Those 3 guys were hugely instrumental in really keeping the Simunauts going on these techniques because they are food scientists. Our students are not quite trained in some of these techniques that they were using. And some of the companies are companies, and there were companies from all over the world. This is Colonel Deltec, who's using microfood production that is rich in protein, fiber, and essential nutrients that are fed this patented media, I think, to form foods that can then be used in space. Nolux is this company that is using artificial photosynthesis to produce plants and fungi, which I think is hugely innovative. And I think we're going to hear some of that tomorrow or Saturday from someone here. And then Interstellar Lab, which is using a nutrient closed-loop system to break down and scale up food production from food waste and plant waste products. Some of the work that was done in our department included food safety, and this is one of our Simunats, Sakura Sajiyama, who's a student in food science. Sensory testing, where we were measuring how likable these foods are. If you think about someone who is captive on a space station, they're going to get tired of eating the same old astronaut food, and so we want to make sure there's a plentiful palette of different stimuli for taste. And finally, I'm going to leave you with, if we don't integrate our systems together, collaborating, working together with all of the technologies that we have now between AI, machine learning, molecular biology, omics of every type, lipidomics, metabolomics, et cetera, we can't really address our food needs for the next century, I think. And this is just the food system map that all of you are familiar with, requiring sustainability, machine learning, circular economy, and upscaling. And I'll stop there. I want to acknowledge my research group, who's quite large right now, with quite a few excellent doctoral students and directed by Dr. Cole and several full-time staff members. These are our collaborators, and I'll point out that Genevieve Sparagna is our lipidomics person for cardiolipin work, and John Newman, Brett Goodpaster and Brian Clark are sarcopenia and diabetologists who do a lot of work in the area of clinical medicine there, and Kedron Baskin and Aine, who are oxygen consumption measurements for cellular energetics, and then Aine, who works with us on our statistics. And of course, our participants, who we couldn't do this without them. So I want to thank you for letting me come be a part of your forum. I'm honored and delighted, and I'll take questions if I have time.